Module 2, Part 2. In the first part of the Module 2, we talked about structures of membranes, movement of lipids within membranes, proteins within membranes, and the movement of proteins within membranes. Now I want to talk about the transport of small molecules across membranes, which is also critical for the life of the cell. First, I will give you a brief overview of the topic and talk about diffusion across plasma membrane without the involvement of any transport proteins. Cells can move substances across membranes by a number of processes. They can use passive diffusion, which does not depend on existence of any protein. Then there are transport-mediated movements of substance across membranes. The characteristic of the transport is usually that protein transporters are involved. There are three subclasses of those. There are facilitated diffusion, active transport, and coupled transport. And finally, there is bulk movement across membranes, which is the movement of very large proteins and is usually achieved by vesicle budding and fusion. This is an overview of membrane transport proteins. Cellular membranes are not only barriers but also conduits for selectively transporting molecules and ions from one side of the membrane to the other. The transport proteins form a protein-lined pathway across the membrane that enables hydrophilic substances to move through the membrane without contacting its hydrophobic interior. There are three main classes of membrane proteins that transport molecules and ions across cellular membranes. Each concentration of the substances also create what is known as a gradient across the membrane. The gradient across the membrane is indicated by the pink triangles with the tip pointing towards the lower concentration, electric potential, or both. It could be in a reverse orientation and depends on the substance that we are talking about. The first group of proteins are called channels and facilitates movement of specific ions or water down their electrochemical gradient. They can be non-gated, open all the time, or they could be gated, open only in response to specific chemical or electrical signals. The second type of transporter proteins are three types, and they facilitate movement of specific small molecules or ions. The uniporters, which is also a facilitated transporter, transport a single type of molecule down its concentration gradient. The other two types of transporters are called cotransporters and known as either sympoters or antipoters. The energy available from the movement of one or more ions down the electrochemical gradient drives the movement of one molecule against its concentration gradient. This is also known as secondary active transport. The third type is ATP-powered pumps, and the energy released by ATP hydrolysis drives movement of specific ions or small molecules against their electrochemical gradient. The table shows the four mechanisms by which small molecules and ions are transported across cellular membranes, and pretty simple and self-explanatory. Some substances move readily and rapidly across the membranes by passive diffusion. In this case, these substances are moving down their concentration gradient from high concentration to low concentration, so no energy is required. There are only a small group of substances that can cross biological membrane, and these include the gases as shown in here. Small uncharged polar molecule such as ethanol to some extent, water, and urea can cross membranes, although not very easily by passive diffusion. So these substances will move from a region of higher concentration through the lipid bilayer to a region of low concentration on the other side of the lipid bilayer. The rate-limiting step is the movement through the hydrophobic core of the biological membrane. So to summarize, the rate of transport via passive diffusion is dependent on three variables. First, it is proportional to the molecule concentration gradient across the bilayer. Second, the size of the molecule, smaller molecules are more permeable than the larger ones. And the third and most important is the molecule's hydrophobicity. The hydrophobicity of a molecule is related to the molecule's partition coefficient, which is K, the equilibrium constant for its partition between oil and water. In other words, the K, or equilibrium constant, determines whether the substance is more soluble in lipid or in water. Substances with higher partition coefficient is more soluble in lipid, say vitamin A, which is lipid soluble. In contrast, vitamin C has a low partition coefficient and is more soluble in water. So vitamin A, being more lipid soluble, will pass through the membrane more readily compared to vitamin C. So a number of substances do not satisfy the three mentioned criteria, and passive diffusion is not sufficient for their transport. So for example, large uncharged polar molecules such as water, glucose, and many other sugars, vitamins like B or C, many large ions, and charged polar molecules such as amino acids, ATP, and say phosphorylated sugar like glucose 6-phosphate, these do not readily pass through biological membranes by passive diffusion, but usually require the help of protein transporters. Now I want to talk about the transport of small molecules across membranes, which is also critical for the life of the cell. So, this is the second type of transport, which is facilitated diffusion. Certain amino acids and nucleosides and some sugars can be transported across biological membranes by this process. In this case, the substances are moving from the area of high concentration to low concentration. So it is still diffusion and still no energy is required. The difference is that there is the assistance of some kind of protein transporter in the membrane. These transporters are usually referred to as uniporters because they can move one type of substance or one class of substance. So here are the differences between facilitated diffusion and passive diffusion. First, the rate of movement for facilitated diffusion is faster than passive diffusion. And that is because the rate-limiting step has been removed. Instead of the substances moving through the hydrophobic core of the lipid bilayer, the substances is actually moving through the transport complex. You have removed the rate-limiting step, so it is faster. Facilitated diffusion is also specific, which means these uniporters only move one substance or one class of substances, and the facilitated diffusion occurs in a limited region of the cell where uniporters reside. So here is an example of an uniporter called GLUT1. GLUT1 is a well-characterized transporter that moves glucose into most mammalian cells by facilitated diffusion. This is a really interesting transporter because it can take on two conformations. There is a conformation in which the glucose binding site is facing outside of the cell. It is outward facing and shown here in 1 and 2. And the other conformation, the glucose binding site, is facing inward toward the cytosol in 3 and 4. Again, glucose is moving down from the area of high concentration to an area of low concentration on this side of the membrane. So this is how this transporter works. Glucose from the extracellular space will bind to the glucose binding site. When glucose binds, this causes a conformational change in the transporter protein, and the glucose binding site is now facing inward. So now the glucose is within the core of the transporter and it can diffuse into the cytosol. Once glucose is in the cytosol, it is often immediately converted to glucose 6-phosphate by an enzyme. This maintains the glucose gradient across the membrane, so it maintains the functionality of GLUT1. Once the glucose is in the cytosol, the transporter can change conformation again, such that the glucose binding site is now outward facing once again. Now, if for a reason this gradient changed or glucose concentration was high in the cytosol and low in the extracellular space, this transporter will work in reverse. Here on the left is a structural model of the human GLUT1 uniporter protein in an inward open conformation towards the cytosolic face. You can appreciate from the previous part that this protein has 12 transmembrane alpha helical segments organized into amino terminal and the carboxy terminal domains, each of which consists of a pair of 3 transmembrane alpha helices. Cut open view of the structure shows the surface electrostatic potential in yellow and blue. The hydrophobic amino acids are in yellow, and you can see that they are around the hydrophobic core of the lipid bilayer, and the hydrophilic blue amino acids line the central channel and present on the exterior and interior of the cytosolic aqueous environment. On the right is a GLUT1 working model cycle known as alternating axis model. In the step 1, the outward open conformation binds glucose. Binding depends on the Km and glucose concentration. Upon binding of the glucose, the transporter changes conformation as in step 2 to the ligand-bound occluded conformation, and then again it changes conformation to deliver the glucose into the cytoplasm, and the release of the glucose is dependent upon the Km and the glucose concentration as well, like step 1. The step 4 is a ligand-free occluded conformation. If the concentration of glucose is higher inside the cell than outside, the cycle will work in reverse and will cause net movement of glucose out of the cell. To prevent that, the cell can phosphorylate glucose to glucose 6-phosphate, which will not be transported by the GLUT transporters. This is to maximize the glucose gradient and uptake of glucose. The human genome encodes at least 14 highly homologous GLUT proteins, GLUT1 through GLUT14, with different Kms. For example, GLUT2 in the liver has a Km of 20 millimolar, increases uptake after a meal when blood glucose is 10 millimolar, and release glucose when glucose blood level drops to 5 millimolar or below. Insulin regulates insertion, removal of GLUT4 into fat and muscle cell plasma membranes, internalized vesicle to regulate glucose uptake. Cellular uptake of glucose mediated by GLUT proteins exhibits simple enzyme kinetics. So here what facilitated diffusion of glucose looks like compared to the passive diffusion of glucose across biological membranes. On the y-axis is the initial rate of glucose uptake, and on the x-axis you have the increasing concentration of glucose. You can see that the passive diffusion is very inefficient mechanism for moving glucose across the plasma membrane. But you can see if you add GLUT1 or GLUT2, which both function in a similar fashion with different rates, of course, you can get much more efficient transport of glucose across the biological membranes. Of course, this kind of experiment would have been done with liposomes and GLUT1 and GLUT2 embedded in the membrane or the bilayer of the liposome. Please understand that in physiological condition, glucose will not be transported into the cell by passive diffusion. It is too large, and experiment is described only to compare the efficiency of the facilitated diffusion. So let's take a look at the experiment in detail. Please refer to the enzyme kinetics and Michaelis-Menten kinetics from the biochemistry prerequisite course material. So in the experiment, V, the initial rate of glucose uptake plotted against the different glucose concentration in the extracellular medium. Note that the experimental initial concentration of glucose in the cell is always zero, equivalent to the concentration difference across living cell membrane. Vmax depends upon the number of transporters. Doubling the number of transporter doubles the Vmax. The Km, concentration at which the rate of glucose uptake is half maximal, depends on the affinity of the transporter for transported molecule. For say GLUT2, the Km would be like 20 millimolar, has a much lower affinity for glucose than GLUT1, with a Km of 1.5, half the Vmax. The low Km of the GLUT1 Uniporter enables transport glucose into most mammalian cells. Blood glucose is 5 millimolar and GLUT1 transports at 77% of its maximum rate. Let's take a look at the Uniport that distinguish Uniport from simple diffusion. Number one, the rate of substrate movement by uniporters are far higher than the simple diffusion through the phospholipid bilayer. Number two, the transported molecule's partition coefficient K is irrelevant because the molecule never enters the hydrophobic phospholipid bilayer. 3. Vmax depends on the number of uniporters in the membrane, achieved when the concentration gradient across the membrane is very large and each uniporter is working at its maximum rate. 4. Transport is reversible but always down the concentration gradient. Direction of transport will change if the direction of the concentration gradient changes. 5. Transport is specific. Each uniporter transports only a single type of molecule or a single group of closely related molecules. Affinity of transporter for its substrate is the Michaelis constant Km, the concentration of substrate at which it transports half the Vmax. Please note an important correlation. The Km of an enzyme or a transporter here is inversely proportional to its affinity to the substrate. Higher the Km, lower its affinity, and vice versa. Although water is not as large as glucose, but still it is a polar molecule but can't pass through the plasma membrane by passive diffusion. The cells have protein transporter for water known as aquaporin found in all domains of life from bacteria to eukarya. The illustration shows the molecular structure of a bacterial aquaporin. The one shown in the picture is a tetrameric protein having 4 identical subunits. And each of them has a hydrophilic water channel in the center, with the water shown as the blue dot. The outside structure contains hydrophobic amino acids to interact with the phospholipid bilayer. We have seen several porine structure in the previous part. Please remember that porine superfamily of proteins have hundreds of different structure, and although we have seen a few of the beta barrel type of porins in the previous part, the aquaporin shows here is not a beta barrel structure. Instead, each of the subunits are composed of 6 transmembrane alpha helices, as you can see in the colored rod-like structures. Here is a beautiful experiment to show the role of aquaporin. The natural environment for the frog oocyte is water. To prevent water from entering the oocyte, no aquaporin channels are present on the oocyte plasma membrane. But in an experiment, a frog oocyte was microinjected with aquaporin mRNA made in vitro, and the mRNA translates the aquaporin, and the cell inserts the aquaporin on the plasma membrane, its natural destination. The cells below are the control without expression of aquaporin, and the upper row of the cells are expressing aquaporin. The cells were placed from isotonic 0.1 molar salt to hypotonic 0.035 molar salt environment, and within a few minutes, the aquaporin transports water inside the cell from hypotonic environment. The oocytes swell and eventually burst, known as osmotic lysis, a classic experiment to show the role of aquaporin in water transport. The first two transport mechanisms which we talked about, passive diffusion and the facilitated diffusion, are when substances are moved down their concentration gradient. So from areas of high concentration to regions of low concentrations. But in many instances, cells need to move substances against a concentration gradient. This is carried out by active transport, and in this case, energy is required. In these examples, the energy is generated by hydrolyzing ATP. So active transport is carried out by what is known as pumps, which are ATP binding protein complexes that harness the energy from ATP hydrolysis to move substances against their concentration gradient. There are a number of classes of these pumps. There are P-class pumps. The P-class pumps are responsible for moving a variety of ions against their concentration gradient across the biological membrane. They are usually made up of two identical catalytic alpha subunits and two beta subunits. Only one alpha and one beta are shown here. The alpha subunit contains the ATP binding site and hydrolysis region. The alpha subunits are really the workhorse of these pumps. The thinking is that the function of the beta subunits is for the structural integrity to help maintain the interaction between the alpha subunit and the membrane. Another large class of pumps are the V and the F class pumps. We are not going to consider in detail these pumps in this course, but just have a brief overview of their function. In general, both V and F class pumps are responsible for moving hydrogen ions or protons across membranes against a concentration gradient, and they do not have phosphoprotein intermediates like P-class pumps. The V-class pump couple ATP hydrolysis to transport proton against a concentration gradient, and they generate a low pH of plant vacuoles, lysosomes, and other acidic vesicles in animal cells by pumping protons from the cytosol into the organelle or vesicle. The F-class pumps, on the other hand, work in reverse to use the energy in a proton concentration or voltage gradient to drive ATP synthesis in mitochondria and bacteria. The third class of pumps, which are the largest class of pumps, are known as ABC superfamily of active transport pumps. ABC simply stands for ATP binding cassette. The structure of these pumps are very similar within this family. There are usually two large transmembrane regions labeled T and the cytosolic ATP binding and hydrolyzing region and labeled here in A. Each ABC protein is specific for a single substrate or a group of related substrates like ions, sugars, amino acid, phospholipids, cholesterol, peptides, polysaccharides, or proteins. So let's look at a couple of examples of these P and ABC superfamily class of pumps. Please note that we classified ABC proteins as pumps, but ABC transporter is more correct name. Why? Because although the ABC transporters will always require ATP to operate, it may not transport molecules or ions against the concentration gradient, but down the concentration gradient. In that sense, not all, but these ABC transporters are channels and not a pump. An example of this is CFTR channel that we will discuss in a minute. So please remember that ABC transporters are classified with pumps in the sense they always will require the energy from ATP, but can be a channel, means moving substances down the gradient, and not a pump, which moves substances against the gradient. But these are the few exceptions. There are many ABC transporters that move substances up the gradient. Please also note that this is not the case with the other P, V, or F class pumps. They always move substances up the gradient. One pump that is well characterized and found in most animal cells is the sodium-potassium ATPase. It is a member of the P-class pump family. What this pump does is that it moves two potassium ions against a concentration gradient and three sodium ions against a concentration gradient simultaneously. Again, it harnesses the energy from ATP hydrolysis, which is shown here, and you can see this pump has a characteristic 2 alpha subunit and 2 characteristic beta subunits. So let's look at this pump in a little bit more detail. So like GLUT1, this pump takes on two conformations. In this diagram, these conformations are labeled as E1 and E2. In the E1 conformation, there are 3 high-affinity sodium binding sites facing the cytosol and 2 low-affinity potassium binding sites facing the cytosol. So all 5 ion binding sites are facing the cytosol. Because these are high-affinity sodium binding sites, the E1 conformation of this pump readily binds to 3 sodium, and when this happens, there is an ATP hydrolysis event, and one of the aspartate becomes phosphorylated on the transporter itself. When this occurs, there is a conformational change, so all 5 binding sites end up on the outside of the cell. This is the E2 conformation. The E2 conformation, which is characterized by 3 low affinity sodium binding sites on the outside of the cell and 2 high affinity potassium binding sites on the outside of the cell. Because these sites have become low affinity, sodium can diffuse into the extracellular space, and the 2 high affinity potassium binding sites readily capture 2 potassium ions from the extracellular space. And when this happens, there is a hydrolysis event and the aspartate become dephosphorylated. And you get a conformational change back to the E1 conformation. And at this point, the potassium binding sites become low affinity and the potassium ions diffuse into the cytosol. This is a very active pump in most animal cells. Again, 3 sodium ions are pumped out of the cell and 2 potassium ions are pumped into the cell in one cycle. This table shows a typical intracellular and extracellular ion concentration of several ions, and just for an idea, and you do not have to remember those numbers, but what I want you to remember, a very important key fact, true for every single cell in the metazoans. The ATP-driven pumps and channels, which we did not consider here, generate a huge gradient of ions between the cytosol and the surrounding extracellular fluid. Among those, the sodium and the potassium gradient is the most important to maintain many biophysical properties, for example, cellular communication or excitability. The K+ concentration is much higher in the cytosol, maybe 20 to 40 times higher than the extracellular fluid or blood. In contrast, the sodium concentration is about 8 to 12 times lower in the cytosol from the extracellular fluid or blood. So as your take-home message, please remember that potassium concentration is high and sodium concentration is always low inside the cell or cytosol, and the reverse in the extracellular space. Here is an example of the ABC superfamily transporter. The ABC superfamily of membrane transport proteins is the largest family of transporters, and it is found in both prokaryotes and eukaryotes. This particular transporter was originally known as MDR1 transporter, which stands for multi-drug resistant transport protein 1. The relevance of that will become clear in a minute. But today it is known as ABCB1. It was the first ABC transporter that was identified. So what this transporter does, that it is able to take small substances as shown here and flip them on the biological membrane, or flip them from one leaflet to another, which facilitates the biological release of these small substances out of the cell. The ABCB1 transporter is somewhat specific for a single substrate or group of related substrate. It can move ions, amino acids, phospholipids, and even proteins. It can move a family of charged molecules. So this is quite relevant in chemotherapeutic interventions in patients that have cancer. Often, tumors become resistant to chemotherapy after chemotherapy is applied, and this resistance is facilitated by ABCB1. So this is how it works. Here is a cancer cell, and it has an ABCB pump indicated by the blue oval on the plasma membrane. Most chemotherapy agents are small, hydrophobic, or polar drug or toxins. These chemotherapeutic drugs can pass into the cell by passive diffusion because they are usually hydrophobic or non-polar. So they can easily pass through the plasma membrane. They are pumped out of their concentration gradient by the ABCB1. But what happens often in the cancer cell is that there are spontaneous genetic mutations, and sometimes these spontaneous mutations lead to an increase of the ABCB1 pumps, resulting in significant number of pumps in the plasma membrane. So in this case, the cell is strongly expressing ABCB1. The chemotherapeutic agents enter by passive diffusion, but they are rapidly pumped out to the extracellular space. They have no effect. They are not able to kill the cancer cell because a lot of these agents are cell cycle toxins that block cell division. When we talk about cell division, we will talk more about these chemotherapeutic agents. So you will notice that I have resistant in quotation marks. It is not the cells are so much resistant, but the cells have such efficient pumping capabilities because they have so many of these pumps in the plasma membrane that when a chemotherapeutic toxic molecule enters, it is rapidly pumped out. So for all intents and purposes, it is resistant, but not really. So in cell culture, you can add enough of the chemotherapeutic agents to kill cancer cells to overwhelm the large number of the ABCB pumps in the plasma membrane. But in vivo, you can do that because you would have given a patient such high dose of chemotherapeutic drugs, it would be lethal. So for all intents and purposes, these cancer cells are labeled as a resistant to chemotherapeutic agents. Let's see how the ABCB1 binds to these wide variety of hydrophobic and hydrophilic drugs. The illustration shows a multi-drug transporter ABCB1 or MDR1 structure and a model of the ligand export. The upper picture shows a cutout model of ABCB1, the protein bound to two molecules of QZ59SS, a drug analog, as in the black in the structure pocket. The important few points about the ABCB1 transporter is that the serine 289-290 affect the ligand specificity of the transporter. As we mentioned, it is a multi-drug transporter. Many different hydrophobic and hydrophilic molecules can bind in the pocket. If the ligand is hydrophobic, it partitions in the inner membrane leaflet and then enters the binding pocket of the ABCB protein, where it interacts with the hydrophobic aromatic side chains tyrosine, phenylalanine, which are in the yellow portion of the cavity. In contrast, a hydrophilic ligand can directly bind from the cytosol. The binding of the drug triggers the conformational change for ATP hydrolysis, which provides the energy to change conformation to expose the drug toward exoplasmic leaflet of the lipid bilayer. If the drug is hydrophobic, it diffuses into the exoplasmic leaflet and then out of the membrane. A hydrophilic ligand can diffuse directly into the aqueous extracellular medium. Another ABC transporter called ABCB4 has phospholipid flippase activity. Here is an experiment which shows the flippase activity of ABCB4 in vitro by fluorescence quenching assay. Mammalian ABCB4 expressed in a temperature-sensitive yeast mutant localized to intracellular endoplasmic reticulum vesicles in its normal orientation. The cytosolic surface of the vesicles are faces outward when isolated. At first, a synthetic phospholipid contained a fluorescently modified head group, the blue dot, containing lipids were added to the medium containing the purified vesicles, get incorporated primarily into the outer cytosolic leaflet of the vesicles. In step 2, if ATP was added to the medium, ABCB4 flips a small fraction of the outward-facing phospholipid to the inside leaflet. In contrast, the media without any ATP will not flip any phospholipid inward. In step 3, a non-membrane permeating dithionite destroys the fluorescence of fluorescent head groups exposed to the medium. So only the labeled phospholipid flipped into the inner leaflet will preserve fluorescence, and all the ones on the outer leaflet will be quenched, having no fluorescence. So the greater fluorescence or less quenching is observed in the presence of ATP than its absence indicates ABCB4 has flipped some of these labeled phospholipid to the inside leaflet, which were not quenched by dithionite. Step 4 can be used as a control where the addition of detergents to the vesicle generate micelles and make all fluorescent lipids accessible to the quenching agent, and all the fluorescence will be quenched. You can also see the graphical representation of the fluorescence quenching. The table shows selected human ABC proteins. There are more than 50 different mammalian ABC proteins, eukaryotic cell ABC proteins localized to the plasma membrane, intracellular organelles, membranes. They have high expression in liver, intestine, and kidneys, where natural toxic and waste products are removed from the body. So far, we have talked about moving substances across the membrane against the concentration gradient, harvesting the energy that is stored in ATP. There is another way in which cells can move substances across membranes against concentration gradient, and that is through coupled transport. This is also energy consuming because substances are moving against a concentration gradient, but in this case, the cells are exploiting the energy stored in a gradient rather than energy stored in ATP. So coupled transport, as the name suggests, involves the movement of two substances across plasma membrane simultaneously. One of these substances is usually moving down its concentration gradient, and so the energy in that gradient is being harnessed to one of the substance moving against its concentration gradient. So when the substances move in the same direction, the transporter is known as symporter. So once again, for a coupled transport, you need the assistance of some kind of transporter, And when both substances move in the same direction, the transporter is called a symporter. In this example, you have a symporter moving sodium in the direction from the extracellular space to the cytosol down its concentration, and then moving a second substance right here against the concentration gradient, but in the same direction. On the other hand, cells can move substances in the opposite directions. So in this case, the sodium is moving from the extracellular space to the cytosol down its concentration gradient, and this other substance is moving from the cytosol to the extracellular space against its concentration gradient. In this case, the transporter is known as an antiporter. So let's look at one example of a symporter and one example of an antiporter. So here is an example of an antiporter that is very well characterized and found in cardiac muscle cells. There is a sodium-calcium antiporter, and in this case, sodium is moved from the extracellular space into the cytosol down its concentration gradient, and simultaneously, calcium is pumped out of the cell against the concentration gradient. Let me remind you again that P-class pump we talked about before, which moves 3 sodium ions out and 2 potassium ions in. This P-class pump is responsible for maintaining this gradient. So this entire system is actually exploited to treat individuals that are in congestive heart failure in emergency situation. So calcium is really important for the beating of the cardiac muscle cells, and you need relatively higher intracellular calcium concentration in order for cardiac muscle cells to beat. When people have congestive heart failure, the calcium concentration is an issue. So what can happen is an emergency situation when these individuals can be given digoxin or avobane. These are 2 well-characterized pharmaceutical agents, and what they do is inhibit the action of sodium-potassium P-class pump. So when this happens, the sodium concentration increases within the cell, and when that happens, the sodium-calcium antiporter becomes non-functional. The calcium concentration increases in the cardiac muscle cell, and this facilitates the beating of these cardiac muscle cells. This helps individuals who are in congestive heart failure. It helps their heart cells pump. So I think you can appreciate that many of these transporters cooperate with one another. We saw how sodium gradient is so important for the sodium-glucose symporter and sodium-calcium antiporter. The illustration here shows the cooperation between several of these transporters. First, I want you to notice that these epithelial cells are polarized with tight junctions and established biochemically distinct apical and basolateral plasma membrane regions that contain different transport proteins. The differentiation limit glucose transport through this paracellular pathway between the cells. Here is a sodium-potassium P-class pump that maintaining the sodium gradient across the cell membranes. The sodium gradient is then exploited by the sodium-glucose transporter to pump glucose from outside the cell inside the cell. There is a GLUT2 transporter over here that is moving the glucose down its concentration gradient by passive diffusion. So I think you can appreciate how all of these transporters cooperate with one another. Here is another example, which is acidification of the stomach lumen by parietal cells in the gastric lining that happens after any food intake to kill pathogens and to make food acidic for the stomach enzyme function, which requires acidic pH to function. But the parietal cells acidify the stomach with HCl while remaining a neutral cytosolic pH. The apical membrane contains H+, K+ ATPase. The P-class pump generate 1 millionfold of H+ or proton gradient. The chloride channels balance the H+ charge, preserves electroneutrality, means equal plus and minus charges, and there are potassium channels as well to maintain the ion equilibrium in the cytosol. In cytosol, carbonic anhydrase generates bicarbonate from CO2, and excess OH minus maintains the pH of the cell cytosol. In the basolateral membrane, the passive CO2 diffusion from the blood into the cell and the anion antiporter exchanges the bicarbonate and the chloride ions. There are a number of drugs available in the market, many as OTC, that are proton pump inhibitor drugs, inhibit this pump to decrease the stomach acid secretion to treat heartburn and acidity. These transporters are important for the life of the cell and human health because there are a number of genetic diseases or inherited diseases that are associated with mutations in the genes that encode these pumps. And these tables on these next two slides really illustrate how important these pumps are. Let's look at a couple of examples. For example, there are a large class of inherited cardiac arrhythmia that occur due to the mutation in genes that encode potassium pumps, sodium pumps, or calcium pumps. There is another inherited disorder known as the benign familial neonatal convulsions, which result from the non-functional potassium pumps. In myasthenia gravis, which you have heard about, which is a mutation in a gene that encodes a sodium pump. The inherited disease you may have most familiar with is cystic fibrosis. The pathology of cystic fibrosis results from a mutation in a gene known as CFTR, cystic fibrosis transmembrane conductance regulator, that encodes a chloride transporter, and the chloride transporter is a member of ABC superfamily. So let's look at how a mutation in this channel actually contributes to cystic fibrosis. So here are some characteristics of cystic fibrosis. The primary cause for the disease is the production of thick mucus that clogs the lungs, which can lead to infection and pneumonia. There is obstruction of the small bile ducts in the liver, the ducts in the pancreas, the small intestine, vas deferens, and also malfunctioning sweat glands, all due to the production of thick, gluey mucus. Basically, all the small ducts and glands in the body become clogged with thick mucus. Here are the genetics of the cystic fibrosis. It affects mainly Caucasians. It is a recessive trait. 1 in 20 are carriers. 1 in 2,500 children are born with the disease, and currently there are approximately 30,000 people living with cystic fibrosis in the US. So, in the late 80s, there was a major effort to identify the gene that was responsible for cystic fibrosis. In 1989, it was identified as a gene on chromosome 7. The scientists were Francis Collins and Lapchi Siu. Francis Collins was at the University of Michigan when this discovery was made, and Dr. Siu was at the University of Toronto when they collaborated on this project. You may be familiar with Francis Collins. He was the director of National Institute of Health, and he's also directed the Human Genome Project. So here is the structure of the CFTR chloride transporter. Again, it is a member of the ABC superfamily. So it has two characteristic transmembrane domains, two ATP binding sites and hydrolysis regions. It is expressed in the lung, liver, pancreas, intestine, reproductive tract, and skin. So not surprisingly, this is where you see the pathology of the cystic fibrosis. This illustration shows a simple cartoon of the functioning of the cystic fibrosis transmembrane conductance regulator, CFTR. Please note that the ABC cystic fibrosis transmembrane conductance regulator, CFTR, is a chloride channel, not a pump. Means the transporter carries chloride down the concentration gradient and not up like a pump. But even with this down the concentration gradient, ATP or energy is required. The CFTR needs an additional regulatory domain on the cytosolic phase, the yellow R in the figure, and must be phosphorylated, shown as the red P, before the ATP is able to power the channel opening. So the R phosphorylation stimulates the tight binding of one ATP to the A1 domain. Then a second ATP binds to the A2 domain, leading to the formation of a tight intramolecular A1-A2 heterodimer. This opens the Cl- channel. The relatively stable open state becomes destabilized by A2 ATP hydrolysis to ADP and PI and disrupts the A1-A2 dimer interface, leading to channel closing. So this is how the CFTR works in a normal lung and CF lung. There are endothelial cells and line the airway of the lungs, and these produce and secrete a mucus that ends up in the lumen of the airway. So what happens in the normal situation is that the chloride is transported into the lumen through the CFTR protein. So now there is a net negative charge in this mucus that has been secreted by these cells. This then draws sodium across this layer. Sodium ions move between the cells in the layer. This is a type of transport where certain ions can actually pass between the cells, known as paracellular transport. So sodium is drawn across to balance the negative charge, and so now you have a high sodium chloride concentration in this mucus. In response to the high concentration, water is drawn across into the lumen in this area and hydrates the mucus. Water is moving across due to osmotic pressure because of the high sodium concentration in the mucus. But what happens in the CF lung is that there is a point mutation that renders the channel non-functional. So chloride does not move into the lumen. As a result, sodium do not enter the mucus by paracellular transport, and thus there is no increase in sodium chloride, so no water is drawn across. This results in a production of mucus which is very thick. In the case of CF lung, it traps pathogenic microorganisms which can lead to pneumonia. In the case of other small ducts, it causes clogging. So you can see how the malfunction of just one of these pumps, it's a point mutation, it is just one amino acid change, can lead to devastating physiological condition such as cystic fibrosis. Now I want to talk about the transport of larger macromolecular structures and proteins across membranes, which is also critical for the life of the cell. The last form of transport that I just want to mention is shown in this one slide is bulk flow. The reason I'm only mentioning it because it is going to be the subject of the entire next module. So bulk flow is the movement of large fluid droplets or very large particles from the outside of the cell into the inside of the cell, or the movement of proteins from inside the cell to outside of the cell or between organelles within the cell. So here are a couple of examples of the bulk flow. When the cell internalizes very large particles such as bacteria, the process is known as phagocytosis. When the cells move large fluid droplets across the membrane, it is known as pinocytosis or pinocytosis. Sometimes it is known as a fluid phase endocytosis, and that is also an example of bulk flow. There are a number of cells that will secrete protein, so exocytosis or secretion is a form of bulk flow. And then proteins are moved between the organelles within the cell, which is another example of bulk flow. So all bulk flow is mediated by vesicle budding and fusion. So vesicles will bud from one membrane, move to the other membrane, fuse with that membrane, and release their content. This is true for phagocytosis, pinocytosis, where the vesicle bud from the plasma membrane and move into the cell. It is true for the secretion, where the vesicles form inside the cell, fuse with the plasma membrane, and it is true for intracellular transport, where the membrane of one organelle fuse with the membrane of another organelle. So in the next module of this course, we will be focusing on these vesicle trafficking pathways. This concludes part 2 of module 2 and also concludes module 2.